Cool. So this is our second edition of this, which is quite exciting. And just for some context, this came up at a meeting in Crete this year at the Mesenchymal Cells and Health and Disease, where we've started to build a bit of a stromal network. And slowly we've been advertising that, and we just want to try and build a bit of momentum. So this is now the second edition. Chaiman did the first one, and Vasiliki's also now signed up to be our European representative. So yeah, so the paper today is a bit of an oldie. I'm going to call it an oldie but a goodie. I think that I haven't had a lot of chance beyond my PhD lab to discuss this paper, but I think it's quite interesting. There's a lot of data in there, so please stop me along the way if you have any questions. There's actually like so many mouse models. I mean, I don't know if you can see it, but there's like a whole supplementary table of mouse models in here, but they all use kind of similar Cree drivers, so that will be hopefully easy to follow along. All right, so let's get started. I'll have a laser pointer. Just kind of what we've been doing is saying, really the aims of this session is to discuss the paper, be kind and fair to each other, share and discuss ideas, and also just have a little bit of fun as well over the next hour or so. All right, so this is the paper. Actually, I've never met anyone from this paper, actually. So does anyone just nod or don't know? Does anyone know anyone on this authors? Okay, cool. So someday, hopefully, they will turn up at a Stromal conference and we'll be able to ask them more details because there's a few mysteries, I guess, for me still. So as a way of introduction, I think that broadly, the first point I want to make, and I think the authors make, let's move this a little bit out of the way, is that, I'll leave that for that, is that fibroblasts and secondary lymphoid organs are really critical, that they have this immune-like signature. So they're really core fibroblasts from like a myofibroblastic point of view, but they have these unique genes that they express so that they can functionally support immune cells at these sites. So I think, you know, if you're coming to this from not as a secondary lymphoid organ fibroblast biologist, this is a really key distinction to make, that outside of tissues of secondary lymphoid organs, really these are unique fibroblasts essentially. The second aspect to this is that there's been a lot of interest in how they actually form in secondary lymphoid organ sites. In the case of the lymph node, you have early during development the formation of something called the lymph node antigen, where you have lymphoid tissue inducer cells arriving around vasculature, which then recruit mesenchymal lymphoid tissue organizing cells, or LTOs. And there's still actually a lot to be understood about the differentiation and origin of these cells. And this paper back in 2020 made some steps towards understanding that. Just wait for Jamie to join in. Hello, Jamie, welcome. We're just getting started. So please feel free to say hello and also reach out if you have any questions as we go along on this paper. We're just at the introduction stage. So one thing about this paper, so obviously it's focusing on the fibroblast in the lymph node and kind of the concept of what really makes a fibroblast in the lymph node a fibroblast in the lymph node. But one aspect that's interesting and unique from my point of view is that they really look at this key signaling cascade, which is the hippo signaling cascade that converges on YAP TAZ, which eventually lead to impact the transcription factors TEAD. The kind of interesting thing about this paper, and that's why I wanted to know about the authors, is it's really not clear to me why they chose YAP-TAZ. I think at the time and still now, YAP-TAZ is a very interesting candidate in fibroblast biology. There was a paper, I think maybe a year or two ago, looking at cGAS sting signaling in relationship to YAP-TAZ in aged tissues, and that if they reverse this process, you kind of de-age tissues, which was pretty cool. But at the time in 2020, it wasn't really, it almost seems like a bit of a sort of a fishing expedition here of just deleting YAP-TAZ. So it'd be interesting to see what you guys think about that. And I just want to bring up this diagram because I think it's quite helpful. So this paper came out in 2020, and already kind of the ideas around YAP-TAZ as being important regulators in the differentiation of fibroblasts has been integrated. So this is from a review, I think Burkhard Ludwig, who was at the meeting in Crete this year, was involved in this publication. But basically, they're saying that in the early stages of this mesenchymal lymphoid tissue organizer cells, YAP TAZ is a really important factor, and we'll see that throughout the paper to drive this pre-fibroblast like state. And then you really need lymphotoxin signaling and NF-kappa B signaling to drive their formation into a fully functional immune fibroblast. And just to outline that there's still lots of active research into understanding how this kind of classical fibroblastic reticular cell becomes all of its various subtypes that you might see in a variety of papers that include single cell sequencing of fibroblasts in the lymph node. So with that in mind, let's get to how they really started to unpack the role of YAP TAZ in FRC maturation and regulation of lymph node function. So their first kind of, just going to pause. Does that reasonably sound okay to everyone? I've got some nods and thumbs up. Okay, cool. So really in the first figure, what they do is they go for the hammer approach, which is they use the CCL19 CRE, which is quite a popular CRE driver in the field for secondary lymphoid organs because it labels the fibroblasts. And they just decided, let's just constitutively delete YAP TAZ right from birth, essentially. And what they find is kind of interesting, is that you have a reduction in lymph node weights and the cell count, and a reduction in the proportion of fibroblasts. And you can see that in the classic gating here. You've got your protoplanin positive cells, CD31. And so when you delete YAP TAZ right from the start, you're reducing the population of fibroblasts. And they kind of posit a little bit that is due to a reduction in the proliferation. And also what's kind of interesting as well is that not only do you have this reduction in fibroblasts, you start to lose the key compartmentalization of the tissue. So you can see here in the wild type condition, you usually have this nice distinction between the T cell zone and the B cell follicle. But now this is really completely lost. And if you're sort of clued up on your FRC biology, there's known chemokines that are important for this kind of compartmentalization. And so they look at these by mRNA, and CCL19, CCL21, IL-7, and BAF are all reduced by mRNA expression in this context. And also, I think this is geometric mean or something like that. They have this reduction, you can see. Interestingly, CXCL13 is not reduced, and CCL27. But really, the key take home here is that if you get rid of YAP TAZ signaling early on, you start to have these defects. The lymph node still forms, but you have these defects in the compartments. And just to say, I thought this was quite important as well, is that individually on their own, if you delete YAP or TAZ, there's no clear impact, but you really need this dual deletion of YAP and TAZ to actually have some impact on the tissue. And you can see here as well, it looks kind of reduced in size. Let's give a look over here. So what they do as well, so you have this slight lymphopenia, but they don't want us to test, if we challenge the system with an immune challenge, how do the cells respond? And what they found, which I thought was kind of interesting, is that these mice are kind of immune compromised in some way. So they have this reduction in body weight after challenge. I think this is the flu challenge, but and also their CD8 T cells have reduced interferon gamma expression. But from my point of view, you have this such striking difference in kind of the organization, yet you still do have a little bit of immune control. And also in the CD4 T cells, there's really no change. So it seems to be that you have this sort of lymphopenia, you still have some architecture of the lymph node, but it's really specifically your CD8 T cell priming or induction that is perturbed. And I think that for me, this figure 1 would have been nice to have a little bit more on that. So is it cell division and things like that in this context? But does still suggest some robustness. Yeah, Jamie?
Do you know if there was a change in the CD8 total fraction in figure M? Because you see this decrease in IF and gamma positive CD8s from 6.2 to 1.8%, but then they don't show the CD8 fraction itself. Maybe it's in the supplemental.
Yeah, so I think it's in the supplement. I'll just have a look here. So that's the thymus. Yeah, so they don't actually do that distinction. So I think that this is here, right? So you see this percentage.
Yeah,
So they
but
don't go on to look at number, right? So that would be helpful. I think that's sort of my one thought here, is that if they could have a bit more on kind of enumeration of these cell types, maybe it's the effector cells, the memory cells, for example, that might be nice. I also think as well, positioning of dendritic cells, so some imaging of DCs would have been good too, to see if they're mislocalized or something like antigen presentation is disrupted. Yes, so you're right. That is, I think, a slight limitation in this early part of the study.
Yeah.
That's a good point. Okay, so now, but the thing is though, irrespective of this, and the reason why I say I'm surprised there's not more striking differences is when in one of the supplementary figure 6, I thought this was really interesting. So usually fibroblasts are known at steady state to produce extracellular matrix and wrap around some ECM and form these conduits in the lymph node that help drain lymph throughout the tissue so it can be sampled for antigens by APCs. But what was really interesting is that clearly when you remove YAP-TAZ here, so this is now using a reporter for TD tomato for the fibroblasts, and then they've got this ER-TR7, which I think can't remember exactly the collagen it marks, but from the paper that came out a few years ago. But anyway, the point is that the FRCs seem to kind of uncouple themselves from the underlying conduit. And usually you might expect, with the main function of fibroblasts is to kind of maintain conduit integrity, and that if you unwrap from the conduit, surely you'd get lots of leakage of lymph from this conduit. But what they find actually is that despite this unwrapping, if you do a FITC tracer tag, you don't really get any change in the kind of leakage from the conduit. So this is kind of hidden in figure 6, and it's quite interesting is that somehow the integrity of conduit flow is maintained independent of fibroblasts wrapping around. So I think there's actually a lot here to unpack, where why is it that when you delete YAP-TAZ, you uncouple yourself from the matrix? Is it just because they're becoming more contractile, but yet can't remodel their matrix? Is there a change in the way the cells adhere to each other? There's actually quite a lot of questions here. So I thought this was very interesting, and probably not an overlooked aspect, but I think something that would require further investigation. So then I think probably they were pleased with this finding, but maybe they wanted to do a bit more and see if they can disrupt the lymph node a bit more. And so in the pathway of signaling, in the Hippo pathway, you have LATs kinases, which like negatively regulate YAP-TAZ. So if you delete LATs, you basically overactivate or hyperactivate the activity of YAP-TAZ, so that moves to the nucleus to induce transcription factors. So in this next experiment, what they do is they say, okay, well, the deleted YAP-TAZ has some effects, but what happens if we really ramp up YAP-TAZ signaling by removing the inhibitor? And this is where they start to get some really crazy stuff. So this is, again, this is not inducible. This is right from throughout differentiation, essentially. And what they find is the lymph nodes are really hypercontracted and disrupted. Again, they do some kind of classic looking at lymph node weight, total cell count, percentage of fibroblasts, and they just validate here that in their model, you really get this nuclear localized YAP. So that means you've activated YAP and it's moving to the nucleus to do its job. But what's different here as well is that somehow this causes neonatal lethality in many of the animals, and then they're much smaller as well. I think they don't really go on to say like how that is possible, but the important finding here is really that you have a complete suppression of kind of your normal formation of the lymph node. And I think really in this case, and the reason why they go on to do inducible stuff later, is that it's due to the failure in forming the kind of the primordial lymph node antigen. So in the wild type, see at E18.5, you basically have the recruitment of LTI cells, and then these lymphoid tissue organizing cells that arrive, and then there's this positive feedback loop to kind of start expanding the emerging lymph node and recruiting immune cells. But in the LATS1-2 knockout, this is really failing, and so you essentially get a failed lymph node construction, basically. But the key thing is that, like, why is that the case? And I think really what they're saying is that you have these hypercontractile, hypermyofibroblastic-like fibroblasts forming, and And you can see that here with classical markers, right? So you would all be aware of, alpha SMA, collagen 4, CD140 beta, and you can see there's the loss of these key kind of distinctive markers like CCL19, CCL21 that would mark a kind of healthy and normal fibroblastic reticular cell. Again, here's some more markers that we know are really important. So IL-7, ICAM, VCAM, RADCAM, they're all changed when you hyperactivate YAP-TAZ. Now, they do try and finish this figure in an interesting way. They then just say, okay, well, what happens if we get rid of the entire signaling pathway of YAP-TAZ? And whilst they say, they do say a partial rescue, and it's somewhat true, but the lymph node still makes its architecture, but it really isn't completely rescued. I mean, they're still quite lymphopenic, they're quite smaller. So I think you do need YAP-TAZ signaling to some extent throughout the entire development of the lymph node. But really, if it's overactivated, you really suppress this transition into a normal fibroblast and kind of good fibroblast differentiation, let's say. So with that in mind, I think they then wanted to change gears and move to more kind of deletion in the adult tissue. And I suspect this is probably the first experiment they did, because I'm pretty sure this is, this is when they use the CD140 beta cree instead of the CCL19. So I suspect they probably started with this and then they went back to CCL19. And that's because it was a relatively null finding. So when you delete YAP-TAZ now in the adult tissues, so this is after the lymph nodes form normally, but now you provide tamoxifen and you get rid of YAP-TAZ signaling, there's really no effect. So that's quite interesting, actually. So if you don't have YAP-TAZ at all in the adult tissue under homeostasis, at least, there's no difference. And sort of a bit of an aside here, which was somewhat rounded, but also could be quite interesting, is that we know that protoplanin is a really important marker for fibroblasts, especially in the lymph nodes. We also know that protoplanin does increase in expression during inflammation. And what they show is that YAP-TAZ signaling can alter the accessibility of the protoplanin promoter. And they did this by ATAC-seq. But what was kind of curious, again, this was hidden in the supplementary. And I tried to figure out where they did this attack, like what cells they did it in, but to me it sounds like the way I've read it is they've done this in skin LGR5 positive stem cells. So I really don't know why they didn't try this in fibroblasts. This was a bit confusing to me, but at least it's an aside to suggest that potentially YAP-TAZ is impacting protoplanin, the key marker, but it's not sufficiently altered when you just delete YAP-TAZ itself. Now they think, okay, well, probably oh crap, that's not very good. But then they went to their LATS-TAZ, LATS1 and 2 inducible deletions. So now you get hyperactivation of YAP-TAZ in the adult tissue only. And this is where they start to see some more striking effects, which is that when you do this in the adult tissue, you really, again, drive nuclear localization of YAP. But now you're basically driving a myofibroblastic a phenotype again. So again, they use the classic markers, alpha SMA, collagen 4, CD140 beta, and protoplanin, and just show this really kind of hypercontractile-like network. And you can see, even in the cross-section here, that you've basically forcibly shrunk the lymph node. You lose all of the key marker genes, such as CCL19, 21, IL-7 back. And probably this lymphopenia, but I suspect as well there's like some contractility-induced kind of effects here as well. And importantly, when you lose all these markers, you get a whole bunch of defects, which kind of alluded to, which is that if you provide immune cell transfer, you really get a failure in the localization of these immune cells. You can see here, both B cells and T cells really fail to like access and get into the lymph node. So there's really a migratory defect, but also if you provide OT1 CD4 T cells and challenge the animal over, you also get a antigen-specific defect in division. And you can see that here with this CSFE dilution graph. So you get good division in the wild type condition, but in an inducible that's 1-2, you really fail to induce the division of these T cells. And again, at the slight aside here, they kind of looked at the endothelial cells, because I think they wanted to say in this example that it's really just the failure to get in because of signals from the fibroblasts, and that the endothelial cells are healthy or normal. They do kind of look quite normal, but there might be potentially some spacing defects or some weird things also going on with endothelial cells, because there is an important relationship between the fibroblasts and the endothelial cells in these HEV regions. But that's just a small aside. All right. So I think it's somewhat unsurprising, but they did start to look at what are the key genes associated with this transformation of the fibroblasts in the induced TAZ 1-2 model. And clearly there's a downregulation of nice signaling of like good chemokines, but then they see this kind of classic myofibroblast signature. So you've got TGF beta, you've got lots of actin, you've got lots of actin remodeling genes coming up. So I just thought I'd put this here for completeness. We can also discuss it a little bit as well, because it's kind of interesting that they really are becoming classic myofibroblasts, and you'd see loads of these genes in kind of calf environments too. Oh yes, that's this was also quite interesting, I thought, as an aside, is that there's something really inherent about the scaling of the lymph node tissue. So that is, if you look at cell counts in this model, like you get a massive reduction, but somehow still there's a proportional decrease, right? So you get a contraction of the lymph node. So you still have some T and B cells, and they are proportionally relevant to the size of the tissue. So I think that's pretty cool, even though you do this kind of crazy manipulation of the tissue. All right, so maybe if I go a couple slides back, they saw that there was also potentially a relationship with lymph toxin beta receptor, which I mentioned earlier in the talk has been shown to be quite important for this final push towards immune fibroblast-like states. And so what they decided to do is take a model that's been used in the literature already, which is this lymph toxin beta receptor deletion, and validate whether there's any relationship to the phenotype in that model to YAP-TAZ. And what they found, they kind of recaptured known features of this model, which is that when you delete the receptor, you no longer get lymphotoxin signaling, and so you kind of return again to this myofibroblastic state, where you've got this alpha SMA collagen 4 high. But what was quite interesting in this model, which hadn't been looked at before, is that when you delete the receptor, you get this translocation of both YAP and TAZ to the nucleus, suggesting that there's likely to be some signaling relationship between these two signaling cascades. So in sort of, they're kind of building towards, at this moment in the paper, towards that lymphotoxin signaling and YAP-TAZ signaling are linked. But before they get there, they do this slight aside, which I think is potentially a bit unnecessary, and it's only a minor criticism, but still one nonetheless, is that they wanted to somehow prove that the kind of change in the fibroblasts was a cell intrinsic one because of YAP-TAZ. And the experiment they do is really kind of almost obvious, right? So they take the fibroblasts out of their model and they induce the hyperactivation of YAP-TAZ, which they've already kind of shown does change cells into myofibroblasts in vivo. So I don't know why they decide then to do this in vitro. I guess they wanted to show through these mutant forms that you can also drive the formation of these myofibroblasts in vitro. But I think they'd already sufficiently proven that if you dysregulate specifically in the fibroblasts the signaling of YAP-TAZ, then you get these myofibroblasts forming. So I don't know why they have to do it here again, but they then potentially use that to set up that you can take out primary cultured fibroblasts from human and do the same in human. So when you apply these constructs where you have constitutively active YAP or TAZ, you then drive again these kind of myofibroblastic like phenotypes. So that's pretty cool. You can in human cells see similar effects. So here you just have low SMA, and this is increased, and that's quantified down here. I guess it's 2 dots, so maybe it was a reviewer question or something. So who knows? Okay, so this next figure is quite a lot of biochemistry, I guess you can say, and I thought it was quite nice. So they really, at this point in the paper, that I would say I'm pretty convinced that YAP and TAZ are really important regulators of fibroblast function, both during development and in the adult tissue. But they wanted to understand the interplay between lymphotoxin and YAP TAZ itself. And so the first thing is that when you so the downstream effectors of lymphotoxin signaling are P52, RelB, and P100. So they wanted to check, if I co-IP YAP TAZ, do any of these proteins kind of stick together? And they find that P52 does. They then go on to look at when they have this hyperactivated form of, or phosphorylated form of TAZ, do you also find the binding of these lymphotoxin proteins? And when they put their inputs in, they do find that specifically when you have this hyperactivated form, you get the binding of P52 and RelB. But then they kind of wanted to say, well, are they both binding at the same time or at different sites? And they start to go towards this through some mutagenesis. They find that when they have this mutant of this PPY motif, they actually have lose binding of P52. But this is where it's a bit tricky, is that when they look at this next domain, which is a WW domain, which they predict RelB will bind to separately, they don't really, I guess, when they delete this, you kind of lose P52, but still have RelB. So I think they, in the end, never really can confirm that LB is binding specifically to YAP TAZ, but certainly P52 is via this domain. So I think there's still a little bit for them to unpack. I guess they summarize here in how these two molecules bind and whether that's actually relevant for then their downstream signaling here. I guess they follow that up a little bit using a cell line, a hex cell line, and do this assay, which is quite interesting. They take their CCL19 promoter and put luciferase after it, and just ask, if we add P52 or YAP or TAZ, does this start driving the promotion of CCL19, which is known to be really important for the final step into fibroblast, or the FRCs becoming this immune, fully mature immune fibroblast. And it's quite a nice experiment. So when they add P52, they can see already just by putting that into the hex cells, that starts to drive the expression of CCL19 and luciferase. But when you have this mutant, which doesn't have the CCL19, that's kind of their background control. When you add TAZ on its own, it doesn't actually do anything. When you have both P52 and TAZ, you have this quite striking upregulation of CCL19. So it suggests there's this a distinct relationship between signaling in lymphotoxin and P52, and that you need both of these proteins present before you can start to get activation of CCL19. And they kind of show that here in this last experiment, where you have without the agonist, you have lots of TAZ signaling, and so then you have when you have the oh wait, which one is that? That seems the wrong way around. Anyway, when you have the agonist, you then lose this expression of CCL19. But I think they go some way to building this diagram, which suggests that when you have engagement of lymphotoxin, you have presumably suppression of YAP TAZ translocation. No, wait, it's probably the other way around here. But nonetheless, what they show is that these two signaling pathways are tightly interlinked and are important for driving CCL19, which gives them the rationale to start perturbing both these pathways to really see what happens in vivo again. And this is kind of the final figure, which is very interesting because there's been a lot of discussion about where these lymphotoxin, these LTO, mesenchymal LTO cells arise from. And so what they do here is they have their wild type, then they have your YAP TAZ deletion constituted from birth, and same with the lymphotoxin receptor. And then when you get rid of all of these signaling pathways, you start to see these perilipin positive aggregates forming essentially throughout the entire tissue. I think this is very interesting. And then same in the inducible. So if you do this right from birth or in the adult tissue, you start to have this formation of like adipocytes. And what's also really interesting is that it seems like this is only the case for sort of skin draining lymph nodes, so the inguinal, axillary, brachial, and I think this is the mesenteric. So somehow, if you get rid of all of this signaling, you start to get essentially adipocytes. And the kind of last experiment here, which was interesting, is you can really see that by the genes. So in this inducible deletion, when you delete in vitro, you can see this increase in expression of kind of adipocyte genes. And I haven't got here a figure, but they also took some kind of stem-like cells and push the cells towards this adipocyte lineage or towards fibroblast lineage when they perturb YAP/TAZ or lymphotoxin. So then they really finish off with this nice summary, which I think is quite helpful, where really in the steady state lymph node, you need a nice balance of YAP/TAZ. So you really don't want overactive and you don't want none at all. If you push the cells towards higher YAP/TAZ, you get this myofibroblastic FRC-like phenotype. Now, I will say this, whilst true, is an extreme case in their data, right? They've completely hyperactivated YAP/TAZ. So it would be really interesting to see, in a normal inflammatory condition or in other conditions where you have slight changes in YAP/TAZ, what the fibroblasts start to look like. If you get rid of YAP/TAZ completely, you actually just lose fibroblast growth, and that if you get rid of lymphotoxin signaling as well, you have adipocyte transformation. So I thought it was a really nice paper to explore these. I'm going to finish it with a few discussion points, and that is, I think they are pretty keen to point out that YAP/TAZ loss is separate to overactivation, which kind of obvious, I guess. So the YAP/TAZ loss is really to do with fibroblast maintenance and proliferation, but the LATS1/2 loss and overactivation is really driving a myofibroblast phenotype. There's still some work to do on the kind of the signaling part of it, and maybe they could have done some stuff in vivo, but it was pretty good. Like I said, I think we know that YAP/TAZ is a mechanosensor, so it would have been really nice to see, in different mechanical conditions, how fibroblasts have their YAP/TAZ translocated. That would be really nice. And also, their readout was kind of these changes, but what about the transcription factors like TEAD, which is the end effector of YAP/TAZ? I wonder if they had a TEAD knockout, would there be a similar effect? And whether that's an important question to follow up on. Also, in this final figure, there are still some fibroblasts, so it would have been nice for them to phenotype that a bit. And also in the field, this idea around this PI16, this CD34 positivity, which suggests these kind of precursors to generating fibroblasts. Are these cells positive for that? What is their relationship to those type of adventitial reticular cells? And I also want to say as well that I think the lymph node is actually quite robust. Even with these changes, you still have some architecture in the lymph node and aggregation of immune cells. So that's pretty interesting. So I'll leave that there for now, and I'm happy to sort of chat through some things and discuss. Let's see. That's all good, Jamie. Cool. All right. What do you reckon? There's heaps of data in that paper.
Yeah, that's first of all, excellent presentation. I don't do anything with lymph nodes, so the fact that I understood the whole thing is
Okay,
awesome.
that's good.
Um, I was wondering if, like, what you think about, I remember we were talking about this actually at the conference where you were saying how lymph nodes can undergo like these huge expansions or I guess like responses to like infection and then they go back to
Mhm,
their original size.
mhm.
Do you think this YAP/TAZ mediated myofibroblast phenotype, where you suddenly have all these like myofibroblast like fibroblasts, I guess? Do
Mhm.
you think it could revert back to a more FRC kind of homeostatic condition?
Yeah, I think that's a good point. That's why in this graph I was saying that
Yeah.
um, really they're only in reality their main phenotype came from hyperactivating the cells.
Yeah.
So, and I think it's unlikely that you're going to have this kind of condition of all of your fibroblasts with nuclear translocated YAP in a normal inflammatory response. That being said, it may be that over time the cells do just have YAP TAZ translocating more easily to the nucleus, and then you start building up fibrosis, and that's why over time you're not restoring homeostasis as easily. And I think the idea is that that CGAS Sting YAP TAZ paper goes some ways towards that, where if you modulate YAP TAZ in these adult tissues, you can kind of restore them to like a young phenotype. So yeah, probably that you have in resolution, you have to also balance YAP TAZ, because if you don't resolve kind of any imbalance there induced by inflammation, maybe you don't get back to the steady state. And there is a paper out there that published back to back with my paper that did look at YAP TAZ throughout the early phases of inflammation. So it goes nuclear localized and it comes back down. So there is already some evidence towards that, which is nice. I also I have started to look at this a little bit in the context of calf formation as well. Not in the primary tumor, but in the metastatic lymph node. So I presented some of this yesterday, that if you get close to the border of an invading metastasis, you have this dysregulation in YAP TAZ, where you have a lot more nuclear localized. So it is likely that this is a very important factor in kind of chronic or kind of damage-based impacts on fibroblasts, basically, which I think they would get to, or I'm surprised this lab hasn't followed up more on a study, because they clearly have all the tools.
Yeah, they have some awesome mouse models.
Yeah, it's kind of wild, actually. Because also, I think in the year also after this, Shannon Turley's lab published on piezo-1, which is another mechanosensor, but they kind of look more in pairs patches. So there's certainly, I think, in each kind of secondary lymphoid organ, mechanical signals are very, very key in balancing the normal function. And so that's why I like this paper as well, because it kind of goes some ways towards that. Yeah.
So I also have nothing to do with versus and lymph nodes, and kudos to you for presenting it in a way that was actually very very concise, because it was a hell of a lot of mouse models in this one. And I really liked your comment on what's happening with the remaining fibroblasts there, because
Yeah.
reading it, of course, not when it was if I read it, like, I don't know, a couple of years ago, maybe I wouldn't have had the same question, but it kept popping up in my mind of what is going on with these remaining fibroblasts and what is actually their identity now,
Yeah.
especially in this, universal precursor, paradigm that we have.
Yes.
So I thought that was really interesting, and I'm glad you mentioned it.
Yeah, I think that certainly is like, yeah, Matt's paper, Matt Bucher, I can't remember how to say his second name. Yeah, with all the PI16, yeah, cross tissue, fibroblast stuff. Yeah, definitely be interesting. I think also as well, if I go back to it, right at the start, let's escape that. I think still, to me at least, and maybe I haven't read up enough, this kind of idea that, let's click through to it. Also, they talked about how the lymph node antigen couldn't form, but it's really not clear to me why the mesenteric lymphoid tissue organizer cells could not be recruited properly. I think they didn't really elaborate too much on, if you delete or if you overexpress that's 1 and 2, you get this hyperactivation and this failure in forming the lymph node antigen. I didn't dive too much deeper into why that actually was. And like you said, maybe it could be that the fibroblasts that remain are just really dysfunctional, can't do anything at all, or because there's lots more of these cells stuck in this kind of adipocyte lineage, maybe they just can't do anything. Basically, they really, really require YAP/TAZ to move past this checkpoint and start to become mesenchymal fibroblasts.
And also another thing that Jamin mentioned about you know, these drastic changes in size that lymph nodes go through, you know, infection and stuff. I wonder if YAP/TAZ has a role in, you know, bringing things back to an original size or homeostatic size anyway.
Yeah.
Because now, as we saw here, even though a complete anyway, structural deregulation occurs, the proportion of the populations in these deregulated lymph nodes remains somewhat the same. So I'm wondering if some kind of, you know, maintenance of these proportional numbers is something important enough to be preserved, you know, regardless of.
Yes.
So that was really interesting to me, and I'm glad you mentioned it in you know, your presentation, because it was like a super small figure, you know.
Yeah, I think this kind of in the background is this ongoing theme about, you know, what lymph nodes are like unitary structures, and especially in the mouse, they're very distinct, but in humans, you have many, many lobes. So I think you have to scale your secondary lymphoid organs in an appropriate manner as your organism gets larger. So I think that if you have this nice adaptive effect where your tissues just kind of contract and enlarge based on your kind of requirements for your immune cells, that's kind of the idea. And that the fibroblasts underpin that is also important. I guess the tricky part is that often if you have this lymphopenia or you have, you know, excessive, or you start to trap and proliferate immune cells, the fibroblasts do take a little while to catch up. So sometimes you then kind of have this imbalance where you have immune cells expanding and your fibroblasts are like, oh my gosh, like we need to start proliferating and supporting these immune cells. Then when the immune cells go away, the fibroblasts kind of still are proliferating or at a high number, and then they've somehow got to contract. And I think that still very little is known about the resolution phase, and also after you've had the resolution, and then what happens during secondary infection? Do you have less expansion of fibroblasts? Is it a different challenge model? There are some papers out there, certainly from the lab I'm in at the moment. Scott published some stuff like this a few years ago, but really the mechanisms driving resolution, the mechanisms driving responses to secondary infection is not clear. All good, no dramas. It's nice for you to join. Thank you.
Yeah. I have maybe one last question before we end it
Yeah.
off, unless anyone else
Yeah,
has any more burning questions.
Yeah, it's good.
Do you think I think it was like a couple, maybe it was figure one where
Yeah.
it was the lymph node size looks smaller with deletion of YAP/TAZ, like kind of. But then yeah, I was interested on your take on, is LYVE is a lymphatic vessel a marker?
Oh, yes.
Am I
That's a good
correct?
point. Yeah.
Yeah,
Yeah.
and so I was wondering if you think this has an, like
Mm,
if the lymphatic vessel, I
that's a
actually
good
just don't know
point.
how they're created. Are they created by the fibroblasts, or fibroblasts kind of come in, and do they help support like immune cell
Oh, yeah.
circulation
Yeah,
with the lymphatic vessels? I actually
you're
just don't know. It's
definitely,
a,
you're
yeah,
yeah.
curious to hear.
Yeah. Yeah, no, you're definitely testing my knowledge there in terms of, I probably should read up again on this. So I'm pretty sure the lymphatics during the antigen formation come and wrap around like this and kind of just develop and wrap around. But I'd have to read up and find some references for you. But certainly, I guess I can't answer your question directly, but something that I do find interesting in this paper is that when they stain for YAP/TAZ, I'm surprised it doesn't mark all of the cells because I've used the antibody and it does stain the endothelium as well. And so it would be interesting to follow up to see if YAP/TAZ in the endothelium, both the blood and the lymphatics, is important for their development too. And also T cells express YAP/TAZ. And that's why when I saw this paper, a few years ago, I was like, oh great, at least I can use this antibody for fibroblast. But there's lots and lots of papers now looking at YAP/TAZ in T cells and how they're mechanoresponsive. So yeah, I think that there's still a lot to do in terms of what's the whole mechanical picture, or the whole picture of YAP/TAZ in the tissue as a whole. It seems like fibroblasts in this case are very sensitive to YAP/TAZ manipulation. But it would be interesting, like I said, if you did the same experiment in the endothelium, would there be as much disruption? Or is it quite unique that the fibroblasts rely on YAP/TAZ for their fate commitment and differentiation into fibroblasts? So it's a very good point. Great. Well, that's been really fun. I'm glad that we finally got to do the second one. And thank you all for joining. So I think we actually do, I know Vasiliki, you said you like, and Christina, I think your colleague, to do the next version. So we tentatively have a date, but I think what we'll do is we'll advertise that again in the WhatsApp. And also just to say, I'm currently at the Australian Society for Immunology, and yesterday we had the Stromal workshop. So hopefully you'll start to see some more Australian stromal biologists joining in, which is cool. And also just wanted to say thanks Alexa and Jamie for coming along. Really appreciate that. And as we go along, hopefully we can build some more steam, get some more people coming, and yeah, get this sort of stromal network growing.